The core engineering paradox of biological flight comes down to a simple, brutal constraint. How do you generate the massive power needed to fly without burdening the extremities with heavy, drag-inducing muscle? This animated diagram illustrates the avian solution: strict weight management through mass centralization. Notice how 25 to 35% of a flying bird's total body mass is concentrated right here in the pectoral region, while the extremities hold under 2%. Keeping the wingtips and toes nearly devoid of muscle is an absolute requirement. If the extremities were heavy, the energetic cost of rapidly swinging the wings back and forth during continuous flight strokes would instantly exhaust the animal. The macroscopic anatomy of a bird is an exercise in ruthless weight centralization, optimizing the entire chassis to achieve maximum aerodynamic efficiency. Because the major muscles are packed tightly near the center of gravity, birds rely on a remote control architecture to drive movement at distant joints. Instead of heavy muscle, long, silvery white tendons extend down the limbs. These avascular cables slide efficiently inside specialized sheaths, carrying mechanical force out to the wingtips and toes without adding unnecessary mass. We can see this remote control strategy at work in the leading edge of the wing, which is controlled by a specific muscle called the propetogialis. As the wing joint is extended, the propetogialis mechanism engages, creating an automatic passive tensioning straight across that leading edge. This passive tension holds the crucial airfoil shape steady against the rushing wind. It does this entirely mechanically, requiring zero continuous neural signaling or active muscular energy expenditure. By automating these critical aerodynamic stability functions, the avian skeleton conserves immense amounts of metabolic energy, reserving it exclusively for the primary flight muscles. Those primary flight muscles form the massive pectoral complex, anchoring directly to a prominent blade-like bone called the sternal keel. Because this tissue holds so much total mass, veterinarians use it as the standard palpation site to score a bird's overall body condition. Two distinct muscle groups layer over this keel. The large superficial pectoralis on the outside powers the forceful downstroke, while the deeper supracoracoideus, hidden underneath, powers the upstroke. Lifting the wing from below requires a unique biomechanical detour. The tendon of the supracoracoideus routes upward from the chest, passes through a bony loop in the shoulder called the triosseal canal, and attaches to the dorsal surface of the humerus. As the underlying muscle contracts, the tendon slides straight through the bony triosseal canal, pulling the humerus upward from a fixed pivot point. This rope and pulley arrangement solves a difficult physics problem. It allows the heavy muscle powering the upward stroke to remain safely anchored underneath the bird, maintaining a highly stable, low center of gravity. But macroscopic levers and pulleys are only half the mechanical equation. To understand avian endurance, we have to scale down into the microscopic layout of the individual muscle bundles and fibers. Deep inside these fibers lies the sarcomere, the absolute smallest functional unit of muscle contraction. This diagram outlines the sarcomere structure. In panels B and C, the unit is bordered by Z-bands with overlapping thick myosin and thin actin filaments. Contraction begins when myosin heads bind directly to actin, aggressively pushing it toward the center to shorten the muscle. How quickly and sustainably those sarcomeres fire depends heavily on their fiber typology. Migratory birds rely on type 1 slow oxidative fibers for endurance, while domestic poultry are dominated by type 2b fast glycolytic fibers for quick, short bursts of activity. For a duck or a goose to sustain a migration covering thousands of miles, their type 1 fibers must perform continuous lipid beta oxidation, steadily burning fat to generate the ATP needed to keep the myosin head cycling. Sustaining that oxidation requires a massive delivery of oxygen. To supply the flight muscles, the avian heart maintains an extreme stroke volume, sometimes reaching over 1,000 beats per minute during flight. Macroscopic flight endurance and physical power are entirely dictated by this microscopic fiber composition and the strict vascular network required to keep it running. In modern commercial agriculture, the priorities shift entirely from endurance to yield. Genetic selection aggressively targets those type 2b fast glycolytic fibers in the pectoralis, rapidly scaling their size to produce more breast meat. This rapid post-hatch vessel growth is driven by satellite cells. These are specialized adult stem cells residing on the outer periphery of the muscle fibers, and they are solely responsible for generating new tissue after the bird hatches. Watch as these satellite cells divide and fuse into the existing tubular muscle fibers, donating their nuclei to drive aggressive, rapid tissue expansion. But this intense scaling creates a severe physiological bottleneck. The genetic drive for muscle hypertrophy pushes the tissue to expand so fast that it completely outpaces the growth of the surrounding vascular capillary beds. Without a sufficient blood supply to clear waste or deliver oxygen, the rapidly expanding muscle tissue begins to choke, suffering from severe localized hypoxia and massive lactic acid buildup. Artificially accelerating the sheer mass of the muscle breaks the delicate metabolic and vascular balance established by evolutionary biology. That chronic hypoxia and metabolic stress trigger a destructive cascade. The oxygen-starved muscle fibers begin to die off in a process of tissue necrosis and are often replaced by fat in a condition called lipidosis. In commercial flocks, this systemic cellular failure presents macroscopically as a series of emerging meat myopathies, commonly known as white striping, wooden breast, and spaghetti breast. This photograph shows the physical presentation of these three myopathies side by side. On the left, fat accumulation creates white striations tracking along the muscle fibers. Moving right, you see the progressive degradation of the connective tissue, leaving the fibers completely separated. Inside the bird, the body's extracellular matrix attempts to repair this widespread necrosis. It triggers a self-perpetuating fibrotic response, massively overproducing type 3 fibrillar collagen to patch the dying tissue. These Mason trichrome-stained histological slides show the extent of the scarring. Notice the sharp contrast between the healthy tissue on the left and the fibrotic tissue on the right. In the affected panels, healthy endomysial collagen is completely replaced by disorganized, densely packed scar tissue. This density drives the extreme physical stiffening seen in wooden breast. As the animal ages, the normally flexible divalent bonds within the collagen mature into rigid, non-reversible hydroxylpyridinoline crosslinks, hardening the breast meat entirely. The centralized avian flight engine is a masterpiece of biological engineering, but pushing its structural and vascular limits through aggressive artificial selection results in a catastrophic collapse of tissue integrity.